We will move on to our second subtopic, which is genomics. Genomics is the study of all of a person's genes, which is the genome, including interactions of those genes with each other and with the person's environment. An organism's complete set of DNA is called its genome, and every single cell in the body contains a complete copy of the 3 billion DNA base pairs or letters that make up the human genome. With its 4-letter language, DNA contains the information needed to build the entire human body. And a gene refers to the unit of DNA that carries the instructions to make a specific protein or a set of proteins. And each of the 20,000 to 25,000 genes in the human genome codes for an average of 3 proteins. And located on 23 pairs of chromosomes packed into a nucleus of a human cell, genes direct the production of proteins with the help of enzymes and messenger molecules. An enzyme will copy the information into the gene's DNA into a molecule called as mRNA, and the mRNA travels out of the nucleus into the cell cytoplasm, where the mRNA is read by a tiny molecular machine called ribosome. And the information is used to link together small molecules called amino acids in the right order to form a specific protein. And proteins make up body structures like organs and tissue, as well as control chemical reactions and carry signals between cells. And if a cell's DNA is mutated, an abnormal protein can be produced which can disrupt the body's usual process, which lead to disease such as cancer. Genomics is very well known to be linked to the Human Genome Project. This project, which was led at the National Institutes of Health by the National Human Genome Research Institute US, produced a very high quality version of the human genome sequence that is freely available in public databases. And this international project was a success, completed in April 2003, was under budget and more than 2 years ahead of schedule. And the sequence is not of one person, but is a composite derived from several individuals. Therefore, it is a representative or generic sequence. To ensure anonymity of the DNA donors, more blood samples were collected from volunteers than were used, and no names were attached to the samples that were analyzed. So not even the donors know whether their samples were actually used or not. And this project was designed to generate a resource that could be used for a broad range of biomedical studies. And one such use is to look for genetic variations that increase risk of specific diseases, such as cancer, or to look for a type of genetic mutations frequently seen in cancerous cells. So you can see here, in the early days, this is the DNA sequencing lab, okay, back in 1994, and by 2006, DNA sequencing requires actually much less manpower. So this is a chart of composition of the human genome. As you can see, the mammalian genomes have around 3 gigabase pair. 1 gigabase pair equals to 109 base pairs, but only 1.5% codes for proteins. And the repetitive sequences account for half of the human genome. And identifying genes unambiguously is difficult in genomes with large amounts of non-coding DNA. Okay. We have long terminal repeats, or LTR, here in the graph. Okay. This one comprises around 8% of our human genome and is believed to play some role in the integration of viral DNA into host genome and they are mostly found on retroviruses and also transposons. We also have signs here, okay, which are shorter interspersed elements 90 to 500 base pairs in length, and for example, allo sequence, which is about 300 base pairs in length. And these allo sequences are unique to humans and the most frequent human signs. And named for the restriction enzyme ALU, which cuts at AGCT, it is found commonly in repeats. And it is named for the bacterium Atrobacter luteus, from which the restriction enzyme comes. Okay. And then we also have lines, which are up to 7,000 base pair in length, containing transposable elements. Okay, I will explain on this later. That makes RNA coding for reverse transcriptase. And this enzyme can make cDNA from RNA, which can reintegrate into another site. Lines are found in dark G bands of banded chromosomes, and these G bands are rich in A, T, and therefore it has less CpG islands. CpG islands are common in housekeeping genes in the promoters, and they have fewer genes. The Y chromosome has more lines than the X chromosome, and the XS has more than the autosomes. There is less meiotic recombination where there are line elements. A transposable element, TE, transposon, or what we call as jumping gene, is a DNA sequence that can change its position within a genome, sometimes creating or reversing mutations and changing the cell's genetic identity and genome size. And transposition often results in the duplication of the same genetic material. TE makes up a large fraction of the genome and are responsible for much of the mass of DNA in eukaryotic cell. There are at least two classes of TEs. Class 1 or retrotransposomes generally function through reverse transcription, and class 2 TEs or DNA transposons, which encode the protein transposase. Okay. And this protein required for insertion and excision, and some of these TEs also encode other proteins as well. Retro elements comprise 90% of the 3 million transposable elements present in the human genome. And retro elements are split into two large groups, the non-LTR and also the LTR elements. Okay, so you can see here the non-LTR and also the LTR elements. Two of the non-LTR members are present in extremely high copy numbers in mammalian germline, which is the signs with the prominent ALU and MIR repeats, and the long terminal interspersed elements lines containing autonomous L1 and L2 sequences. Signs have no protein coding capacity, and it depends on line elements for amplification. The LTR class elements make up 8% of human chromosomes, and it includes retrotransposons, endogenous retroviruses, and also repeat elements with HERV origin, such as sign R. Endogenous retroviruses are mobile genetic elements, and they are very difficult to be distinguished from infectious or exogenous retroviruses at the time of insertion in the host DNA. For example, the HERV or human endogenous retroviruses. HERVs and related genetic elements occupy around 8% of human genome and are thought to be remnants of multiple ancient retroviral infections. HERV insertions occur in the ancestral germ cell lineage, fixed in the genome and became inheritable. In human DNA, herpes are represented by around 500 groups, including around 770,000 individual fragments, and the individual herpes copies are frequently interrupted by other sequences, such as transposable elements, and can represent 2 or more genetic fragments. 98% of the genome is transcribed into RNAs, and the non-coding RNAs are playing important roles in just about everything the cell does. Okay, these include snRNAs or short nuclear RNAs, snRNAs or short nucleolar RNAs, which is located within the nucleus, microRNAs, siRNAs or short interfering RNAs. These ones can modify the activity of protein coding genes. Typically, these sections of junk DNA come about through transposition, or movement of sections of DNA to different positions in the genome. So as a result, most of these regions can contain multiple copies of transposons. Okay. So these are actually sequences that literally copy or cut themselves out of one part of the genome, and then they reinsert themselves somewhere else. Metagenomics is defined as the study of total genomic DNA obtained directly from environmental samples. And this is actually one of a chapter of genomics. Okay? So the environmental samples include soil, seawater, desert, without actually culturing the microbes in the lab. Metagenomics, therefore, can help to study the microbes that are uncultivable by standard culture techniques, and this will allow the discovery of novel microbial species. And this makes metagenomics an important tool for the understanding of microbial communities. Metagenomics can also be used to understand the evolution, ecology, and metabolism of microbes present in ecological niche, as well as to investigate the relationship between microbial diversity and also ecosystem functioning. And it can be performed with two different NGS-based approaches, or next generation sequencing based approaches, which is the targeted metagenomics and also shotgun metagenomics. In targeted metagenomics, used for the profiling of microbial communities, this can analyze a specific genomic region mostly through PCR-based direct sequencing. And generally, the small and large subunits of rRNA genes are used as markers for identification of microbial species. Shotgun metagenomics provides information regarding microbes present in the community as well as the biological function of the community. So it is based on random shearing of the total DNA into multiple short sequences and then followed by sequencing and reconstruction into consensus sequence.